Progress is impossible without change, and those who cannot change their minds cannot change anything. Hi all, Dr. Clark here again, and we're going to continue talking about principles of science in the environmental science course. We left off, we talked about how the scientific method works, and we went through the example of cowpox and the being immune or developing immunity to smallpox, okay, based on Jenner's research. We talked a little bit about field science and how it can give you a lot of information and can be very powerful. However, setting up controls or dealing with natural disasters and things like that makes it very difficult to do some of those things. Now we're going to not really shift gears, but we're still going to talk about experimental design, and we're going to talk about biases. Okay? So when we talk about biases, we need to basically understand that humans are naturally biased. Okay? You're naturally biased to things that are familiar to you. You're naturally biased to things that you like. You're naturally biased to things that you want. Okay? Now, bias, it's hard to remove all biases out of every experiment. Okay? From things like, let's say we're marking a population of rodents. Okay, so we have caught a bunch of pocket mice. Okay, and we might look at these later, but we caught a bunch of pocket mice in traps, and then we're marking the individuals a certain color. Well, maybe our bias comes in and we say, all right, well, because I want to be able to look at these individuals running around after I let them go, I need to choose colors that can also tell me something about the individual. Like if I paint the back of these pocket mice, if I paint the boys, all of them, we're going to paint them blue. And the girls, the females, we're going to paint all of them pink. And then the juveniles, or the young, that we can't determine whether they're male or female, maybe we're going to paint all of them yellow, or yellow. Okay, that would be perfectly fine. Okay, well, we just may have created a bias. Okay? Because you have to think about, what do predators in a ecosystem, what are they looking for? They're looking for anomalies. They're looking for things that look like the individual might be hurt, bleeding, things like that. So now that you've painted all the females pink, okay, or like a reddish color, maybe that pink or that red looks like blood to a predator. You've just destroyed your experiment based on something that you thought was super innocent and would help you as the experimenter. These are biases. Okay? What you really should have done to eliminate biases is you should have a series of colors, maybe of 5, 6, 7 different colors, and you should randomize them and choose them at random. Okay? And then when you capture new individuals, you should choose the colors at random again. And make sure that your marking technique, the color that you paint the back of the individual, is not having an effect on your population. Okay? There's all kinds of biases. When we do human research, okay, you'll have biases of individuals. You'll have volunteers. You might say, okay, I need 10 volunteers. Okay? Well, if they know that they're going to volunteer for an experiment, there will be a few of the individuals that already want to see the experiment fail. So they'll do things to make the experiment fail. And that's just a bias that people have. Maybe they're not interested in the topic. Maybe they're not interested in the experimenter. Maybe they don't like, a piece of the experiment, so they sabotage it. And these are biases that, experimenters and experimentees have. Okay. So the best way to do that is to increase your sample size and try to eliminate biases from the experiment by having a good experimental design. Okay. One of the ways that you can do this is blind experiments. So you don't allow the individuals who are being experimented on, or what we typically would call the experimentees, you don't allow them to know if they're in the control group or if they're in an experimental group. They're just in a group. So if it's a new medicine, they don't know if they're getting a sugar pill or the medicine, or a really low dose of the medicine, a really high dose of the medicine. They don't know. So as a individual, a experimentee, you come in, you take the medicine, then you write how you feel, what was the side effects, etc. If a bunch of individuals who took sugar pills or the placebo come back and they say, oh yeah, man, that really worked. I feel so much better. I feel like my chest cold is gone, and everything's good. My senses are heightened. Then you know that these individuals that are taking sugar pills are biasing your experiment. And that's why having those good experimental designs are so important. Okay? So what I just described to you is what we call the placebo effect. There's many, many examples of this, from, classic examples of college professors, that have given students, that are 21, given them water that is dyed like it was alcohol, like it was beer, okay, and given it to them in maybe a bar setting, and had the students drinking just water that has been food colored to look just like beer, and the hop smell is there, the beer smell is there, given that to all their students, okay, and then start taking notes. How many of the students act as if they're intoxicated? Okay? And you might be surprised, but some of this research coming out of Harvard shows that 75 to 80% of the students that were drinking water acted like they were intoxicated. Okay? In fact, some of them acted like they're so intoxicated, they wouldn't, after having one or two glasses, they wouldn't drive home. Okay, they at the end of the experiment, the professor asked, okay, how many of you guys need a ride home? How many of you guys are too intoxicated to drive after your one glass of alcohol? Okay. And, there's 25% at times that needed that ride home, even though all they did was drink water. Okay. It's called the placebo effect. Okay. And this is why drug companies that are developing new drugs, okay, they have to use sugar pills, or they have to use, a sugar water, or a known, a known placebo to test that placebo effect. Okay. The other way that you can do this is to eliminate the bias that comes from the experimenter. which is often a very strong bias. Let's think about it from a experimental biologist or a new drug that's coming out in and that experimenter has donated 15 years of their life, 20 years of their life to design this new drug that's supposed to work. Maybe it's supposed to work on obesity, okay? And it's supposed to be a fat-burning drug and really help people that are extremely overweight. Okay? So they develop this drug, and it takes them 15, 20 years. You got to imagine 15, 20 years of your life. If that drug doesn't work, that scientist is heartbroken. That scientist is like, I just spent 15, 20 years of my life for nothing. So there could be a huge bias coming from the scientist. Okay? And that's why most drugs are set up in what we call a double-blind experiment, okay? Which means that the scientist doesn't even know which individual got the placebo, which individual got the real drug. Okay? They don't know until the very end of the experiment which group is the control group and which group is the experimental group. These are great studies, and they tell so much about the bias. And I've been part of these studies where you'll have experimental scientists that are sitting there and they're guessing, right before the end of the experiment, they're guessing which group is the placebo, which group is the experimental. And I'd say probably half the time they're wrong. Okay? And, it's not all that fun sitting there and watching someone say, no, you got to be kidding me. This can't be right. These guys were, receiving sugar pills, and they're reporting that they're better, that they're losing weight, that whatever it might be, they have heightened intelligence, they're doing better on exams, and these people are the experimental people are receiving the actual drug we're testing, and they're doing worse. That's why we set up double-blind experiments. Okay? Is to ensure that things actually work. Now, prior to all this, if we're talking about drugs, we're, another one of the main reasons that we test our drugs on things like mice, pigs, okay, rats. Why we have test organisms is to remove some of this bias. Okay? And that's why early experiments will often have what we call an animal model, or an animal is the experimentee. That way you can eliminate a lot of the biases that would be coming from the animals, because mice aren't going to necessarily choose how they feel or how fast they run on a wheel, or how fast they can move through a maze. They're not going to choose it based on some prior knowledge of, oh, I received a sugar pill, or oh, I received an actual drug. Okay? We'll talk a lot more about this because we'll design these in our experiments that we're going to run in class. We're going to design some blind experiments and some double-blind experiments. Okay, now, sometimes by not following good scientific method, by not following good experimental design, you have individuals that are caught committing what we call scientific fraud. Okay? Now, it's not too hard in today's society to catch someone doing scientific fraud. Actually, it happens fairly often early on that we catch people doing scientific fraud. Okay? Scientific fraud is, there's a lot of avenues that this can work. It could just be bias. It could be outright lying or outright changing numbers in your results. It could be selecting individuals based on prior knowledge in an experiment. There are lots of things. But the key point of scientific fraud is, I would say 99% of the time, it is figured out during the peer review process. Right? So what I want to make sure that you guys understand is how scientific publications work. Okay? This is far different than, say, a textbook or a book that you might pick up from a library or order online or whatever it is. Far different than that. Okay? Scientific research or peer-reviewed research is very different. Okay? So that scientist would go through the steps to create their experiment. They would collect all the data, then they would write it up. Then they take that written work and they pass it off to a peer-reviewed committee. That peer-reviewed committee, that scientist doesn't get to choose who they are. And scientists often won't even know who they are. Okay? Sometimes they know who they are after the peer review process, okay? But often you don't even know who reviewed your material. But here's the kicker. The individuals that review the material are experts in that field. So let's say I publish a paper on old growth trees, old growth forests, and I send it in for publication, okay? And they're not giving my paper to a fish biologist. They're not giving my paper to a lion biologist or a mammologist. They're giving my paper to someone who studies old growth trees, or at least studies plant communities. Okay? And so when they read it, they already know how experiments are done. How sound is that scientific method? Is this possible? And often if there's little flaws in the data or things don't make sense, that peer review committee will pick it up and they'll look at it. Okay? So let's say that it gets past the peer review. Let's say I'm so good at fraudulently, designing my results that it's not detectable with the peer review committee. Okay? So the peer reviewers, they pass it and they put it forward and it's published. Now comes another peer review committee. Okay? Now that it's published, typically scientists read other scientists' work to get ideas about how their experiment's going, or ideas about how to design an experiment, or just in general, if I'm publishing on old growth forests, all the other old growth forest biologists, okay, they're like, hey, new publication on old growth forests, let's read it. And then they start chatting, and they start talking, and they start saying, hey, did you see that table? It seems like the data is strange. Okay? So then maybe they run their own experiments. And this is often what happens. They run their own experiments and they look at it and they say, no, this can't be right. Or better yet, this is what often will happen. They'll look at it and they'll say, hey, I need to see the raw numbers, the real numbers. That's what I need to see. So they contact the researcher who published it and say, hey, I want your real data, your raw data that hasn't been analyzed. I need to see that. You send it to them. Now they have all your data and they can see, okay, this is where he's making up data. This is where the results don't mesh with other things. So maybe it's true, maybe your research was that different. Okay. But often what will happen is the peer-reviewed committee or peer-review group of scientists will start trying to replicate your data, replicate your experiment. And if they can't do it, then they call on you and say, all right, no one can replicate your experiment. Something's going on. You have a biased or flawed experiment. You've committed scientific fraud. Okay? Now, it's very costly in time and resources, and it can take quite a bit of time before individuals who commit scientific fraud are caught. But when they do, okay, it can cost them their entire career. And that's why scientists know that, hey, look, it's better to be wrong. It's better to go through 15, 20 years of your life trying to create a drug and have it not work than to falsify the evidence just so you lose your career. And no one will hire you if you committed scientific fraud. Okay? Apart from that, it also creates mistrust from the public, because it's often reported by newspapers like this individual committed scientific fraud, this study is not true, okay, and it creates this mistrust in the science, okay, and it's this age-old saying where people will say, one bad seed can ruin an apple, or one bad seed can ruin something. So you have one person out of the entire scientific community commit scientific fraud, and it can ruin the ideas of, that field for a very long time. A great example of this is an individual by the name of Andrew Wakefield, or I could call him Dr. Andrew Wakefield, but he's been stripped of his doctor status. But in 1998, he was Dr. Andrew Wakefield, okay? And he has a very classic example of scientific fraud. Dr. Wakefield, or I'm just going to call him Andrew Wakefield because he's not a doctor anymore, okay? Andrew Wakefield wanted to show that the measles, mumps, rubella vaccination, or MMR, causes autism in children. Okay? So in 1998, and some of you have probably heard of this before. Some of you may even have parents that believe that vaccinations cause autism. Okay? That's stemming from Andrew Wakefield's work, okay? Because he published a journal article in The Lancet, which is a very high medical journal, okay, that stated that MMRs cause autism in children. Okay. There it is. Okay. This was the publication. Okay. It was published in 1998. Okay. It had been read by quite a few scientists, okay, but it was published in 1998, and by February of 1919, it was published in February of 1998, and by May 1998, it had been retracted. Okay, which means that the article has been removed. Okay, and it's been pulled. Okay, and you can see it's not just Andrew Wakefield's name on this article. There is a bunch of other people that contributed to this article. Okay. Now later, what was found out that some of these individuals were not aware that Andrew Wakefield made up the data. But some of these individuals were also paid by Andrew Wakefield to be part of the study. Okay. So a couple things going on there. Okay. But a lot of other components with the study that were wrong. And we're going to go through and examine this as an assignment, okay? But I just wanted to kind of bring it forward and show you that there are some classic examples of scientific fraud that can ruin and create a lot of mistrust and misinformation because they're out there. And this was literally only out there for a couple months before it was retracted. But news resources got a hold of it. They started publishing news articles, and celebrities got a hold of it, and they started talking about it, etc. And it created a giant movement. Okay. So what that movement did is it created a lot of mistrust in vaccinations. Right? Prior to this, people didn't care. Like, okay, you have to be vaccinated to come to school. There was a very, very, very small minority of people prior to 1998 that would say, okay, well, I don't want my kid to be vaccinated. Some because of religious beliefs, but I mean, 99% of people were like, okay, it's a vaccination. It's a vaccination against measles and mumps and rubella. I mean, who wants measles and mumps or rubella? I mean, no one. So your child got a vaccination and then they could go to school. Well, after this came out, then it was more choice. Okay? The government doesn't get to tell you whether you can be vaccinated or not. It should be your choice to get vaccinations, etc. And so a lot of articles came out, books came out, people started coming out talking against the use of vaccinations, okay? And they were using this study by Andrew Wakefield as evidence against why you shouldn't have your child vaccinated. If vaccines cause autism, you're doing your child more harm than good. Okay? And it really started a movement, and still today there's still a movement. I mean, you can see all these different things that supposedly, based on pseudoscience and individuals that are not using the scientific method, okay, this is claims like vaccines cause seizures, vaccines cause ADHD, vaccines cause autism still, vaccines cause autoimmune disease, on and on and on. Well, most of you probably don't know any of these women in this picture, but one of the main things, one of the really hurtful things to science is when celebrities start using this misinformation on their platform, start using pseudoscience on their platform. Now, I'm not picking on these celebrities, okay? I don't have anything against these celebrities and the movies that they make or whatever they do, but they're not scientists. And most of them can't read a scientific article, or I never have read a scientific article. But they will talk about things like vaccinations and how they cause autism. All three of these individuals have said that on many occasions. However, there has never, ever been a scientific article showing that autism causes, or that vaccinations cause autism. Okay? And so I just want you to keep in mind things that you might see on Facebook, things that you might see even on the news, things that might come about from individuals who are not scientists about science. Often, you should be looking at these at first with a pseudoscience eye. Okay? Are they really talking about science, or are they just talking about a belief they have? I don't have a problem if you want to not vaccinate your children. But the minute you say that vaccinations cause autism, autoimmune disease, or something that has never been supported by science, then I have an issue. It is your right, it is your choice to choose to vaccinate your children. It's also school's choice to not allow your children to come to school if they're not vaccinated. Okay, so the final thing that I want you guys to do is I want you to answer this question. Now that we've talked about vaccinations, I want you to answer this question. You're going to answer it in your journal, okay? But I want you to think about it. Would you get vaccinated for COVID-19? Okay. Why or why not? I want you to think about that. Okay? We've been spending billions of dollars on trying to create a vaccination for COVID-19. Okay? One of the key components is if you don't vaccinate the majority of people, then you can't eradicate a disease. Okay? The reason why we don't have smallpox any longer in developed countries is because everyone gets vaccinated for it. Okay? Although the new non-vaccination movement allows for the chance that people will get smallpox again because they're not vaccinated against it. Okay? Because smallpox can live in animal communities still today. Right? We haven't seen it. We haven't seen an outbreak of it, but we have seen an outbreak of measles in different regions. Okay? And that's one of the things that we used to vaccinate on, but people are choosing not to vaccinate. Okay? So if there's a vaccination for COVID-19, are you going to get it? Okay? As of today, the newest report is that 35% of Americans will not. 35% of Americans will not get vaccinated for COVID, even if it's free. Even if you do not have to pay for it, 35% of Americans will not get the vaccination. Okay? Are you part of that 35%? Why? Okay. We'll talk about this later.